Hello everyone, my name is Tanvi Patel and I am a pre-doc fellow at NHGRI NIH. I am also pursuing a Doctor of Science degree at Bowie State University. Today I am honored to present my work at the International Conference on Computational Science and Computational Intelligence 2024. The title of my presentation is Training Swin Transformer based GAN for high resolution blood cell imaging with limited data. So in the year 2022, Microsoft made a significant contribution to the field of image-to-image generation with the introduction of the model which is known as StyleSwin. So StyleSwin is transformer-based image generative model. This model set a new benchmark for state-of-the-art performance in high-resolution image generation. This model was trained on ImageNet dataset. So ImageNet dataset is a large and more diverse dataset. So when we applied to medical imaging, there are some challenges emerge. And one critical issue is data scarcity, which gives rise to two major problems. First is discriminator overfitting, where discriminator become too specialized to the limited data and struggle to generalize. And the second is training instability, making it difficult to achieve consistent and reliable results. To address these challenges, we propose StyleSwin-ADA, where ADA stands for Adaptive Discriminator Augmentation. This innovative solution extends the capability of StyleSwin by incorporating adaptive augmentation during the training. By doing so, StyleSwin-ADA significantly improved both training stability and overall performance, making it particularly effective for high-resolution medical imaging tasks in data scarce scenario. And for this paper, we use the blood cell data sets, and it has 8 different classes, including basophil, eosinophil, erythroblast, immature granulocyte, lymphocyte, monocyte, neutrophil, and platelets. And on the right side, the bar chart showing data distribution. This dataset is publicly available on Kaggle and we use from that. This dataset is also included in MedMnist version 2 paper and which was published in 2023. So as a pre-process of the data, we take the raw images. So the process of blood cell image segmentation begin with raw image containing the different blood cells as well. The pre-processing steps are applied to improve the image quality. Then by using the segmentation watershed algorithm separates the individual cells from the background and potentially not touching each other, and the segmented images of the main isolated cell are saved for the further analysis. By after extracting the cells, this is the method or the diagram. Through this diagram, I am showing the method which we used. So this figure shows the workflow diagram of the StyleSwin-ADA architecture. The generator which we used, same StyleSwin generator. The generator takes latent vector Z and produces the style vector W through the fully connected layers using adaptive instance normalization for the style control and Swin transformer blocks. The generator architecture progressively upsamples features starting from the learned constant input, applying double attention and multi-layer perceptron at each resolution to produce the high quality images. And generator create fake images and also combine the real images with the ADA, where the ADA stands for Adaptive Discriminator Augmentation, where the augmentation like rotation, scaling, and other transformation are applied to real and generated images before they fed into the discriminator. And using this, we can prevent the discriminator overfitting by giving the discriminator more data using the ADA. And the code for this paper is available on my GitHub link, which is I mentioned it down, and it is available in my GitHub repository. Then let's take an example of monocyte blood cell. Initially, the model predicts monocyte with 51% confidence. However, if an attacker add noise, which is also known um, adversarial perturbation, the prediction can shift incorrectly to another class, such as lymphocyte. This manipulation is called as adversarial attack. And there are two different types of adversarial attack: black box attack and white box attack, where the black box attack uh, attacker has no knowledge of the model, including the parameters, whereas in the white box attack, attacker has full access to the model. To protect against weak adversarial attacks, we train StyleSwin-ADA on a single class. Training the model on a single class helps it focus on um, identifying and generating specific features for that class, making it more robust against adversarial noise. So weak adversarial attacks often exploit vulnerability in multi-class models by confusing class boundaries. By specializing in single class, the model strength is ability to detect and resist adversarial noise, improving its overall reliability. So by using this model, we can also protect the images or model from the adversarial attack, like weak adversarial attack. A training parameter which we use for this model, like there are total around 17,092 images, and the label shape is 8, like 8 different classes. We choose learning rate 0.0025, and we also take batch size 4. The image, like the latent vector size we use is 512, and we train this model on two A100 GPUs. There are two different laws was used by this model: generator loss and discriminator loss. So we train the model with the same layers and architecture which used by the StyleSwin. We add the ADA in that to protect the model from the overfitting. After that, these are the results. The left side is the StyleSwin model training, and the right side is StyleSwin-ADA proposed model training. And here you can see the FID score, fetched inception distance, fluctuate as training progress, which is the sign of the overfitting. Whereas the other side in StyleSwin-ADA, there the training is more stable as compared to the StyleSwin, which is the sign of prevent the overfitting when we train the model on the same data set. So FID is calculate using the real images, the mean value of the real images, and the mean value of the generated images, and the covariance matrix. Then we also compared the result of StyleSwin and StyleSwin-ADA via pair t-test, which showed an overall statistically significant difference, p is equal to 0.0162. Then this is the visual comparison of generated and original images. The left side are the real images and the right side are the fake, which is known as the synthesis images. And these are the different classes of the images, and you can see that the the generated images are look like same as in real images, except the immature granulocyte results, because in immature granulocytes there are different stains, and also the different type of blood cells in that. So because of the mixture, it has the different output as compared to the other blood cell types. Here I displayed two sections, one side showing real images and the another side is showing generated images by the StyleSwin-ADA. Now I'll take 5-second pause to ask you a question. Can you identify which side is real and which side is fake? It should be challenging for the human eye to distinguish, as the generated images is replicate intricate patterns that is typically in medical imaging. So the left side is fake and the right side is real. Now, to study this further, we train the classifier using real images and test that classifier on generated images. The confusion matrix here demonstrate very few misclassification on blood cell imaging, except immature granulocytes. This is likely due to their mixed staining pattern and the presence of multiple other red blood cell types in the immature granulocytes. This result indicates that even for the classifier, it is hard to distinguish the generated images. To gain more clarity and improve the identification of accurate patterns, we plan to explore this study further. To generate a PCA heat map for 4 different components from both original and generated blood cell images, we first apply PCA to original blood cell images to visualize the feature distribution. Then we flat the 2D array of images features and perform PCA on the top 4 components to create heat map. The heat map illustrates how different images, both original and generated, contribute to each principal component. The lighter color represents the higher contribution, while darker colors indicate the lower contribution. The objective is to assess the similarity or differences between the original and generated images based on the principal components. By comparing the heat maps, we can observe the similarity and differences between the original and generated images, particularly in relation to the fourth principal component. The more aligned pattern between the original and generated images, the better the generative model has captured the underlying structure of the original data. So here you can see the fourth component um, feature distribution on original images is almost similar to a StyleSwin-ADA images, but it is totally different from the StyleSwin images. So it is showing that StyleSwin-ADA images are generated better as compared to the StyleSwin images. So the main contribution are StyleSwin-ADA, which is high resolution blood cell image synthesis we can generate, and like we improved the training stability over StyleSwin. We preserved structure and reduced the noise. Then we compare the results using the PCA and FID score, and high accuracy across blood cell types, fewer misclassifications. In future, we want to combine the real and generated images to enhance the classifier accuracy. Then we also want to address the data imbalance problem using the generated images, the accurate generated images. And we also want to generate early disease to identify the disease in earlier stages. just like the leukemia blood cell images. And I would like to thank you to the Bowie State University and the Medical Genomics Unit, NHGRI, NIH. In the Bowie State University, I would like to thank you to Graduate Resource Center and Department of Computer Science. Thank you. I am open for the questions.